Hello and welcome to this talk, an introduction to high content imaging. Before we begin this talk in earnest, I just want to point out that the majority of the information in this presentation was adapted from the operetta manual and application guide that were provided by PerkinElmer, and images used in this presentation were either adapted from sample images provided by PerkinElmer or taken by Ellie using the operetta unless specified. In today's talk, I will cover what high content screening is, give you an overview of the instrument, discuss the acquisition setup as there are some notable differences compared to a normal microscope. I'll then go through some of the image analysis examples and common assays that are used on the operetta. And finally, we will have some brief talks from some of our users. The biggest difference with high content screening compared to a more conventional approach to microscopy is the level of automation. Automated imaging allows the user to image multiple fields of view and therefore many more cells. By keeping the field of view selection identical, this also removes any bias between wells. With all of these images, it wouldn't be possible to manually quantify each cell or field of view, so high content screening will always have a well-integrated system for automated quantification of the data. The advantage of this approach is that it's automated. It provides unbiased imaging, it allows you to perform high throughput analysis, and it allows for assay validation or screening. Of course, there are also downsides, such as the need for careful optimization of sample preparation and image analysis. The volume of data generated can be quite large, and the unbiased approach limits your ability to image specific regions of interest. Super resolution imaging is also restricted when doing high content screening. Now I'm going to give you an overview of the instrument that's available in our platform. It uses a white light based xenon lamp for fluorescent imaging. It has 8 excitation filters and 10 emission filters. It has a 2, 10, 20 and 40 times objective lenses. The detector is a Peltier-cooled CCD camera that's capable of 14-bit images. It can be used for imaging in different modes, such as wide field, spinning disk confocal, transmitted, and digital phase contrast. The system also has a live cell imaging chamber with temperature and CO2 control. And finally, the Harmony acquisition software has integrated image analysis building blocks. Now we are going to discuss the image acquisition setup on the operetta and some tips for sample preparation. A key component of any automated imaging is the ability to autofocus. You will note from the photo of the instrument that it doesn't have a focus wheel. Instead, the bottom of the plate is detected using the reflection of a long wavelength laser from the bottom of the plate. This works a little bit like how a bat or a dolphin uses sonar, but instead it's using light instead of sound. However, the autofocus might fail if the bottom of the plate varies in thickness, and this happens a lot with standard culture plates as they aren't designed for imaging, so you should always use a higher quality plate. Autofocus might also fail when the plate bottom is not flat. For example, if the well is U-shaped, in this case you need to make sure that you're imaging the center of the well. If you try imaging near the well boundary, you may also have issues. This can be fixed by inputting the correct plate geometry into the software. And lastly, if the buffer or media level is too low, this can interfere with the reflection of the autofocusing laser. BOMP has 5 objectives for use on our operetta, only 4 of which can be loaded at any one time. These lenses fit into 2 main categories: long working distance and high numerical aperture. The long working distance lenses have a larger depth of focus, lower resolution and lower transmittance, so they are best used for thick bottomed plates. In contrast, the 20 and 40x high numerical aperture lenses have a shallower depth of focus but higher resolution and light transmittance. These require a thin plate, but the image quality will be much improved. Here are examples of some cells imaged with 5 lenses on the BOMP operetta. Starting with the nuclei, you can see that the magnification increases from left to right. However, when we zoom in on images acquired at 20x, you can see how much the higher numerical aperture improves the image quality. You can also see this effect when we look at the cell body, which is this middle row. The most conclusive demonstration of the importance of choosing the correct lens is when we look at the organelles. Here, the scale has been matched for each objective, and it is really only with the high numerical aperture objectives that we can resolve individual organelles. You may think that we can just use the lens with the highest resolution for each experiment, but the caveat here is that as the magnification goes higher, the field of view will become smaller, so you will end up needing to take more images to grab the same number of cells. At higher magnification, this not only takes longer, but will also generate significantly more data. This image shows you the size of the field of view for each objective. So when choosing the objective lens for your experiment, you also have to consider the minimum number of images or cells required for your quantification. So please check the application guidelines for the suggested number of cells required for your experiment. To give you a practical example, the size of each image is 2 to 3 megabytes regardless of what objective is used. When you multiply this by the number of channels, fields, planes, wells, time points and plates, you can determine the required disk space for your experiment. For example, an experiment with 3 channels imaging all possible fields would be 230 gigabytes at 40x, but only 60 gigabytes at 20x. The operetta accepts a wide range of plates and slides, which gives you more flexibility when designing experiments. These can be standard slides or chamber slides. Of course, the operetta is also compatible with any format of multi-well plate. However, it can also accept non-standard plates. The Harmony software comes pre-configured with plate templates for our common commercially available plate types. It's important that the operetta knows the exact dimensions of your plate because the wrong plate type can cause an autofocus failure. In Harmony, it is also possible to manually create a template if it is not already in the software, and we can help you make a specific template for your experiment. There are a few important considerations when you are selecting a type of plate to use. First, think of the material that makes up the plate bottom. It's recommended that the plate should have a glass bottom or optical grade polystyrene because these have good optical properties. And you should avoid, if you can, polystyrene that's used for tissue culture, as this can have potential autofluorescence. Another thing to consider is the thickness of the plate bottom. So ideally you want a thin one, which is less than 200 microns, because this will have better optical properties and allow you to use a high numerical aperture lens. By contrast, a thick plate bottom, which is greater than 200 microns, can only be used with a long working distance lens. Next we have planarity. So you should always try to have a plate bottom that has thin glass or thick plastic, as these are more likely to have a flat surface. What you don't want to have is thin plastic, which can potentially be bent and cause a focus failure. Finally, you should consider the geometry of your plate. A lower plate bottom is preferred as it allows the objective to get closer to your sample. Here you can see in this diagram the plate height is marked with the H. Avoid, if you can, plates that have a high bottom height and these ribs, as they can restrict imaging positions. If you are unsure what plate is best suited to your experiment, you can contact Hilling-Higgins at PerkinElmer to arrange a sample plate. As mentioned in the instrument overview, the operetta has multiple imaging modes. It has Brightfield, digital phase contrast for easier segmentation, wide field fluorescence, and confocal imaging for thicker samples, or if you need higher signal to noise ratio. Unfortunately, this mode will also be much slower. Starting with Brightfield, advantages are that it is label free and it's non-phototoxic. That makes it ideal for long-term live cell imaging. The downside is that it doesn't allow you to do 3D imaging and segmentation can be difficult. When we use the phenologic machine learning, it can distinguish cells from the background. This can be used to perform general segmentation, but this is challenging, if not impossible, at the individual cell level. An alternative way to segment unlabeled cells is to use digital phase contrast. This uses a computational approach for the generation of phase images based on two defocused Brightfield images. So here you can see the Brightfield image, and if we defocus it and take an upper and lower plane, it allows us to calculate this digital phase contrast image, and you can see that it's going to be much easier to segment individual cells. The advantages of using digital phase contrast are that it's label free, and again, it's non-phototoxic and it's ideal for long-term live cell imaging. However, it also provides a fluorescence-like intensity profile, which provides better cell segmentation, which you can see in this image. One significant drawback is that it's not useful for 3D segmentation, for example, imaging multi-cell layers. The operetta is, of course, capable of fluorescent imaging. As with all fluorescent imaging, the benefits are very specific labeling and the potential for good 3D. The operetta has an inbuilt live cell chamber that is supplied with CO2 and can be heated for optimal cell growth. However, it does not have any way to control humidity. This table shows the rate of evaporation from wells in a 96 well plate after 4 hours. You can see that evaporation is much higher in the edge wells. To counteract this effect, you should always fill empty spaces and wells with water and use a gas permeable plate or seal the top of the plate when you're doing live cell imaging. Here is an example of live cell imaging where a time-lapse video was taken with digital phase contrast. Another use for live cell imaging is measuring cell proliferation in a high throughput manner. Here the plot of cell number over time has been color coded and overlaid on each well. You can see that the rate of proliferation is affected by the well position, and cells seated at the edge have a significantly lower rate of growth compared to the central wells. So this is something to consider if you're doing a live cell experiment. A final note about sample preparation is to adapt your seeding density to suit the type of imaging you will be doing and the subsequent image analysis. So it should be high enough to have statistically meaningful results, but low enough to segment cell areas properly. Generally, the confluency of your sample should be 70 to 90% for most applications, but never 100%, and 40 to 60% for applications if the signal is on the plasma membrane. Other factors to consider when planning the seeding density are the growth rate, the cell line you'll be imaging, the well area, and how long you will be imaging them for. Now I'm going to go through some common assays that are carried out on the operetta. This is not an exhaustive list of the possible applications available on the operetta, but I will go through one example in detail that represents each different class of analysis. The main takeaway point from this list is that many combinations of counting, quantification, and tracking can be applied to cells, organelles, and any other structure. First, I'm going to go through an example of counting the ratio of live to dead cells as a representative example of nuclear analysis. Determining the ratio of live to dead cells is a common metric for assessing the efficacy of cytotoxic compounds. In this example, all nuclei have been labelled with DAPI, or you could use another dye like DRAC5. The sample has also been counterstained with the membrane impermeant dye BOBO3 that can only enter dying cells and label the nucleus when the membrane has been disrupted. These two wells have differing concentrations of the compound of interest, and visually you can see that the well with the higher concentration has more dead cells. To determine the percentage of dead cells, we first need to detect all nuclei using the DAPI signal, and here you can see the outline surrounding each of these nuclei in yellow. Next, we need to measure the intensity of the BOBO3 signal, and using an analysis sequence in Harmony, we can detect where the BOBO3 signal overlaps with the nuclear ROI. And in this way, we can find the BOBO positive cells. Then it's simply a matter of calculating the percentage of dead cells by dividing the number of BOPO3 positive cells by the total number and multiplying by 100. This analysis sequence can be applied to the well plate, so we can test multiple concentrations of cytotoxic compound and then plot this against the percentage of dead cells. Each concentration can then be replicated on a plate depending on the plate format. This data can then be used to determine the EC50 value of your cytotoxic compound. The next group of experiments I will discuss are concerned with the quantification of one stain in relation to another. And in this case, we're going to look at receptor internalization. The aim of this imaging and analysis experiment is to measure the level of G protein coupled receptors activated by the endothelin 1 ligand. So GPCR is a membrane receptor that can bind with this ligand and then concentrates in the perinuclear region after internalization. Here, the nucleus has been labelled in red and the GPCR is labelled green. The first step in this analysis sequence is to define the cell region using the reference stain DRAC5, as this labels the cytoplasm and the nucleus. Other options for measuring individual cells are to use digital phase contrast, as we discussed before, or something like the cell mask dye. If the nuclear and cell area are known, the perinuclear region can then be calculated based on the relative distance between the nucleus and the cell border. In this step, we have now defined three regions within the cell: the nucleus, the cell body, and the perinuclear region. This can also be used to define the cell membrane by calculating the distance from the nucleus to the edge of the cell. In step 2, we're going to use the areas defined in the previous step and measure the mean intensity of endothelin 1 in the perinuclear region and in the whole cell. In step 3, the internalized intensity is going to be calculated by defining a measurement in harmony where the mean intensity of the endothelin 1 signal in the perinuclear region is divided by the mean intensity in the whole cell. This value can then be plotted against the ligand concentration. And finally, we can expand this out to the whole well and compare the levels of endothelin 1 internalization between wells and display this graphically. Next, we're going to cover an example of a morphological measurement and look at measuring cell shape. Cell rounding occurs in many circumstances, such as when cells are dividing, dying, or fail to adhere to a surface. In this experiment, the cell bodies have been labelled with tubulin GFP, and the treated cells have been inhibited from normal cell spreading, which should give them a different morphology to the untreated cells. Using this fluorescent channel, it is relatively simple to detect individual cell bodies using an operetta analysis sequence. And you can see here that in this example, the seeding density is appropriate for the measurements we want to make, as there aren't too many cells and it's possible to perform accurate cell segmentation. There are many ways of quantifying cell morphology, such as measuring the area, roundness, width, and length of an object, or these can be combined to make more advanced measurements like the width to length ratio and width to length ratio when fitted in an ellipse. Here you can see an example of cell bodies with a low width to length ratio, which indicates that they are elongated, whereas cells with a high width to length ratio will be rounder. In this assay, you can see that measuring and comparing the width to length ratio for treated and untreated cells shows a significant difference in morphology. Another common analysis that is carried out on the operetta is spot analysis that looks for regularly shaped round objects. Spot counting and quantification can be used in a variety of different circumstances, such as calculating nuclear fragmentation, fluorescence in situ hybridization, lipid droplet measurements, or tracking organelles. In this example, we want to track phospholipidosis, which is a condition causing excess accumulation of phospholipids in tissues. In this example, it has been induced by a drug and the droplets have been labelled with BODIPI. Spot counting, which looks for smaller, regularly sized shapes, can be used for making morphological and intensity measurements for each lipid droplet, or for making measurements that take into account all spots in a sample, such as the fraction of droplet area. The next two examples cover more kinetic datasets and look at cell migration and tracking. Examples of when imaging migration is useful are tracking wound healing, immune responses, and metastasis. Here is an example of an ORIS pro-cell migration assay that has been carried out with a 2x objective. In this well, the cells have been labelled green and they have been removed from the centre of the well, leaving behind an empty space. You can see that the empty area in the centre is reduced after 24 hours as cells migrate into it. Now I'm going to cover another kinetic dataset and look at cell tracking. Live cell imaging and cell tracking allows you to make kinetic measurements, and it's commonly used for chemotaxis experiments where cells migrate towards a chemoattractant, and chemokinesis assays that study how compounds stimulate or inhibit random movement. In this experiment, the digital phase contrast channel has been used at 10x to image cell movement. Brightfield and digital phase contrast will always be better for this kind of long-term time-lapse imaging, but in this case, digital phase contrast has been used because it allows for much better segmentation, and this makes it much easier to track cells. Here is the same video that was on the previous slide, but now you can see that the cells have been segmented in every frame and they've been color-coded as a way of marking them. This is the same video again, but now we are displaying the track each cell takes as it moves through the field of view. And we can then measure these tracks for things like speed, displacement, accumulated distance, and many more. Here you can see that 2D displacement in the X and Y axes has been quantified for untreated cells and cells treated with cytoclassin D, which is a compound that inhibits actin polymerization. And you can see that in the treated cells that cell motility is impeded and displacement is much lower. If the analysis that you want to carry out is not available in the Harmony software, you can tailor it further by exporting your data and implementing other programs like Fiji or Cell Profiler to do additional analysis. You can see an entire well has been imaged on the operetta and then stitched together using another program. And this image was provided by Marcia Tan here at the university. Thank you for your attention.